Chapter 8, Topic 5, Genetic Engineering. In this topic we're going to be covering different types of genetic engineering, important enzymes in genetic engineering, analyzing DNA, and recombinant DNA technology. The learning objectives for this topic are: be able to explain the importance of restriction endonucleases to genetic engineering, be able to list the steps in the polymerase chain reaction, be able to describe how you can clone a gene into a bacterium. There are three types of genetic engineering that we're going to talk about. There are a lot more of these techniques, but these three are the backbone of genetic engineering. We are going to look at these on the next few slides. The first tool we use in genetic engineering are enzymes known as restriction endonucleases. These enzymes will cut the DNA in specific places. There are a whole bunch of these, and each has a unique sequence they are looking for. Regardless of the actual sequences, all of these sequences are known as palindromes. A palindrome is a sequence that is identical when read from 3' to 5' on one strand, and from 5' to 3' on the complementary strand. We can use these enzymes to open up segments of DNA and add our own genes to the plasmid, or we can use these to cut up the DNA to analyze it. Once we have that DNA cut up, we can run it on a gel. This process is known as gel electrophoresis, and it separates the pieces of DNA by size. If we cut two sequences of DNA with the same restriction enzymes, we can tell how similar the sequences are. You can see an example of this on the gel on this slide. Another important tool that helps us analyze DNA is PCR, which stands for polymerase chain reaction. This tool allows us to amplify small amounts of DNA for electrophoresis or other analysis. Our last tool is recombinant DNA technology. We have talked about this technology throughout this course. The main example of the use of this technology is the ability to have E. coli produce human insulin for use by diabetics. What we have to do to begin this process is isolate the gene. This is a very time-intensive process. Once we have this gene, we can introduce it into a plasmid using restriction enzymes. We can then introduce this plasmid into E. coli cells, and they will begin producing the protein we are interested in. Well, that's it for this topic, and that's it for this chapter. Here's what I would like you to have learned from this topic. Be able to explain the importance of restriction endonucleases to genetic engineering, be able to list the steps in the polymerase chain reaction, be able to describe how you can clone a gene into a bacterium.